Imagine, for just a second, that you are looking at this highly detailed satellite map of a really massive, bustling city.
Right, like Google Earth, but on steroids.
Yeah, exactly. And you can zoom all the way in, and see exactly where everyone lives. You can pinpoint what building they are in, down to the specific floor, even the exact apartment number.
Wow.
But, well, here's the catch. You have absolutely no idea why they moved to that specific apartment.
Ah, I see.
You don't know what their jobs are, and you certainly don't know, who they are gossiping with in the hallway. That highly detailed but utterly contextless map has basically been the frustrating reality of spatial biology for years.
It really has.
I mean, we could see exactly where cells were located in a tissue, but the why, those biological instructions driving them there, was completely missing until now.
Yeah, it is the exact difference between looking at a static photograph of a crowded room and actually understanding the complex hidden social dynamics that dictate who stands next to whom. Without the why, you just have an overcrowded picture.
Yeah. Welcome to this deep dive. Today, we are exploring a truly groundbreaking 2026 paper from the journal Cell, published by Zhang and colleagues, and they introduced this new technology called SPAC-seq and its companion software algorithm, TARDIS.
It's a massive leap forward.
It really is. Our mission today is to understand how these researchers are finally merging CRISPR gene editing with spatial mapping, because they aren't just figuring out what genes do in isolation anymore.
No, not at all. They are uncovering how those genes physically dictate where cells travel, where they settle down, and how they interact inside living tissue.
Okay, let's unpack this. To really appreciate the leap forward here, what is the core hurdle that traditional genetics has been stuck on?
Well, to see the breakthrough, you really have to look at the limitations of traditional CRISPR screens. Now, CRISPR is incredible for figuring out what a gene does.
Right.
Typically, you knock out a gene in millions of cells, you wait to see which ones survive or die, and then you read their DNA. And here is the problem. To extract that DNA, you have to physically grind up the tissue into a cellular soup.
Ugh, a soup.
Yeah, you completely destroy the spatial architecture. You have all the pieces, but no blueprint. And then on the other side of the coin, we have spatial transcriptomics.
Which is the map.
Exactly. This lets us see the intact tissue structure and where specific genes are active, but it doesn't prove cause and effect. It just shows correlation.
So if I'm hearing this right, traditional CRISPR is basically like taking a car engine apart and tossing it into an industrial blender just to see what the spark plugs do.
That is a great way to put it.
You might find the parts in the rubble, but you completely lose the engine. You have no idea how the spark plug actually connects to the piston. So how does this new tech, SPAC-seq, keep the engine intact?
That analogy hits the nail on the head. SPAC-seq, which, by the way, stands for Spatial CRISPR Screen Sequencing.
Yeah.
It solves this by executing the gene edit and the spatial mapping simultaneously right inside the living animal.
Wow, inside the living animal.
Yes. The researchers achieved this by designing a highly specialized retrovirus-based plasmid. They call this plasmid SPAC-seq. They use the retrovirus to infect cells with a massive library of single-guide RNAs, or sgRNAs.
Okay, remind me, what is an sgRNA in this context?
Think of an sgRNA as a homing beacon. It's the exact sequence that tells the CRISPR machinery which specific gene to cut.
Hold on, why use a retrovirus specifically? Because, there are a lot of ways to get CRISPR into a cell.
That is a really crucial detail. So retroviruses are remarkably efficient at permanently integrating their genetic payload directly into the genome of mouse primary immune cells.
And immune cells are tough to edit, right?
Extremely tough. Those specific immune cells are notoriously stubborn. They actively resist taking up foreign DNA. So the retrovirus acts as this highly effective Trojan horse.
Oh, I love that.
Inside this plasmid, they engineered a dual promoter system.
A dual promoter.
Yeah. A promoter is basically the cellular on switch for a gene. The first switch, driven by an enzyme called RNA polymerase III, pumps out the sgRNA to act as the molecular scissors and do the actual gene editing.
Okay, so Paul III makes the scissors.
Right. But, and this is the absolute genius part, they embedded that exact same sgRNA sequence into a messenger RNA molecule driven by a second switch using RNA polymerase II.
Let me see if I'm visualizing this correctly. The cell is basically churning out two identical copies of the sequence. One copy goes off to actually cut the DNA, and the second copy just rides along as a readable barcode that we can easily scan later.
Precisely. It's a built-in tracking tag. Once these cells are infected and edited, the researchers inject them back into a living mouse model.
So the cells are just doing their normal thing now.
Exactly. The cells circulate, they migrate, they attempt to fight tumors, they metastasize, they carry out their actual biological lives. Later, the researchers harvest the tissue, slice it into incredibly thin layers, and lay it onto high-resolution sequencing chips.
Like which ones?
In the study, they used two cutting-edge platforms, Visium HD and StereoSeq.
And here's the magic detail for you listening. SPAC-seq captures both the cell's mRNA, which tells us its current state and what it's doing, and the exact sgRNA barcode, telling us what gene was edited. It captures both of these in their precise spatial location on that glass slide.
Yes, exactly. And depending on the chip, they used two different capturing mechanisms to pull this off.
Okay, lay them on me.
On the Visium HD chip, they used direct capture with splint-ligated probes.
Splint-ligated probes.
Yeah, think of them as highly specific molecular fishing hooks designed to grab the sgRNA barcode directly from the slide.
Okay, fishing hooks for Visium HD. And what about StereoSeq?
On the StereoSeq chip, they use something called mRNA embedding.
What does that mean, mRNA embedding?
So every mature messenger RNA molecule in a cell has what is called a poly-A tail.
Right, I remember that from bio class.
Yeah, it is essentially a long string of A nucleotides added to the end of the RNA. It acts like a protective cap and a molecular shipping tag, basically signaling to the cell that the RNA is ready to be translated into a protein.
Okay.
The StereoSeq chip is covered in these tiny DNA nanoballs that are designed to specifically grab onto those poly-A tails. Because the researchers cleverly embedded the sgRNA barcode right next to the poly-A tail, the chip naturally pulls the barcode down, along with the rest of the mRNA.
That is incredibly elegant. Let me just pause here and synthesize this to make sure we are crystal clear before we move on, because this is dense stuff.
Go for it.
Okay, here's the quick recap. First, traditional CRISPR loses tissue context because you have to grind it up. Second, SPAC-seq edits genes and maps them on a glass slide. And third, it captures both the gene edit and the cell's locations simultaneously. We get the what and the where, keeping our biological car engine totally intact.
That is a perfect summary.
But that brings up a massive logistical problem. If you have a spatial map containing millions of edited cells, that is just an ocean of data. It's just noise. How do you actually find the signal in all that?
The data scale is staggering, you're right. To make sense of this high-dimensional spatial transcriptomic noise, the researchers had to build a completely new software algorithm from the ground up. They named it TARDIS.
Which, as a sci-fi fan, I love. It stands for Target Prioritization Toolkit for Perturbation Data in Spatialomics. It is literally navigating the space and time of cellular data.
It really is.
So how does TARDIS actually comb through millions of cells?
Well, TARDIS operates using two highly distinct mathematical modes, global and local. Let's start with global mode, which looks at the overarching distribution of cells across the entire tissue slice.
Okay.
It relies on a mathematical concept called KL divergence.
Okay, KL divergence. You're going to have to break that down for me without the heavy math.
Sure. Think of a heat map of a massive, crowded sports stadium. Normally, the fans, which represent our control cells, are relatively evenly distributed across all the different seating sections just watching the game.
Makes sense.
KL divergence is an algorithm that compares two different heat maps to see how much they diverge or differ. It basically flags if you suddenly knock out a specific gene and, mysteriously, all the cells with that knockout are clustered exclusively near the stadium exits rather than watching the game.
Oh, I see. But wait, how do you know they didn't just end up by the exits randomly?
That is where the permutation test comes in.
Permutation test.
Yeah, TARDIS takes the data and digitally shuffles the seating chart thousands of times. If the clustering at the exits was just a random fluke, the shuffling would recreate it fairly often by pure chance.
Right.
But if it only happens when that specific gene is missing, the algorithm mathematically proves that the gene edit physically altered where the cells position themselves.
That makes perfect sense for the whole stadium. But what about local mode? Because, a tissue isn't just one big uniform stadium, right? A cell deep in the core of a tumor is dealing with a very different environment than a cell on the outer edge. Does TARDIS account for those microscopic neighborhoods?
It absolutely does, and this is where the math gets incredibly clever. Local mode divides the tissue into micro-niches, these tiny localized neighborhoods. But counting cells in these tiny neighborhoods introduces a major statistical trap called compositional data. It essentially creates a zero-sum game.
A zero-sum game in cell counting. What do you mean by that?
Okay, imagine a room with exactly 100 people. 50 are wearing red shirts and 50 are wearing blue shirts.
Okay, so 50% red, 50% blue.
Exactly. Now suddenly 50 new people wearing green shirts walk into the room. You now have 150 people total. Because of the new green shirts, the percentage of red shirts just dropped to 33%.
Even though the absolute number of people in red shirts never actually changed, there are still exactly 50 of them.
Exactly. It's a mathematical illusion. If TARDIS just looked at raw percentages, an influx of one cell type would make it falsely look like another cell type was disappearing.
Oh, wow. That would ruin the data.
It completely skews it. So to correct this illusion, TARDIS uses a metric called the Aitchison distance and applies a center log ratio transformation.
Okay, that sounds intense.
You can just think of it like an economic exchange rate adjuster.
Oh.
It transforms these biased percentages into a fair, balanced mathematical space, allowing the researchers to know with absolute certainty if edited cells are actually physically moving into a micro niche, or if the math is just playing tricks on them.
That is brilliant. It strips away the illusion entirely.
And it goes one step further.
Oh.
Yeah, what's fascinating here is TARDIS also utilizes a clustering algorithm known as DB-scan.
DB-scan, okay.
This algorithm specifically hunts for local clonal expansions. It scans the tissue to find clusters of cells that have aggressively multiplied together from one single, original edited parent cell.
Oh, so it tracks the family tree, basically.
Yes. It separates a true biological invasion from just a random scattering of disconnected cells.
Okay, so TARDIS has the ability to spot these hidden clonal expansions and correct the math. But what does this actually look like when applied to a living disease? How does an actual tumor exploit these mechanics?
That question leads perfectly into the team's first major experiment. They really wanted to decode the very early stages of tumor metastasis.
Which is when cancer spreads?
Exactly. They took MC38 tumor cells, which is a standard mouse model for colon cancer, and edited them using SPAC-seq with a library of over 1,500 different gene knockouts.
Wow, 1,500 at once.
Yeah. They injected these edited tumors into the bloodstreams of mice, and 14 days later, harvested the lungs to see which tumors successfully seeded and grew.
And when they ran TARDIS on the lung tissue, what stood out?
One gene lit up the dashboard, ICAM1. When they knocked out the ICAM1 gene, those specific tumor cells formed massive, highly aggressive clones in the lungs.
Interesting.
Now, normally, ICAM1 acts as an adhesion molecule on the surface of a cell.
Wait. If it is an adhesion molecule, it is basically a biochemical hook, right? It helps immune cells, like T cells, grab onto the tumor to recognize it. So if the tumor loses that gene...
it drops entirely off the immune system's radar.
Wow.
But SPAC-seq revealed exactly how it drops off the radar spatially. TARDIS showed that these ICAM1 knockout cells weren't just growing randomly anywhere. They were specifically clustering in what we call immune-excluded niches.
Immune-excluded niches, what does that mean?
They mathematically proved these tumor clones were multiplying in physical zones that were virtually devoid of T cells and natural killer cells.
Let's pause and summarize this because the implications are huge.
Wow.
Here is our second mini recap. 1. TARDIS is the software calculating where edited cells move. 2. In tumors, losing the ICAM1 gene helps cancer metastasize. And 3. It does this by creating a spatial safe zone away from immune cells.
Wow.
Here's where it gets really interesting. By altering just one single gene, the tumor cells essentially figured out how to build a localized, invisible shield against the immune system. They hijack their own spatial positioning to intentionally hide from the body's immune assassins.
It is terrifyingly smart.
Think about that for a second. If tumors are actively using spatial geography to hide, can we use SPAC-seq to do the reverse? Can we map out how to help our immune cells breach those shields and go on the offense?
We absolutely can, and the researchers tested exactly that next. For immunotherapy to actually work, CD8-positive T cells, the primary assassins of the immune system, they must physically infiltrate the hostile environment of the tumor bed.
Right, they have to get inside.
The team engineered these T cells with a massive library of knockouts and watched their invasion over time.
So they were basically looking for intrinsic genes that control a T cell's GPS navigation. What did they find?
The standout hit was a gene called CD44.
CD44, okay.
When they knocked out the CD44 gene in the T cells, those cells suddenly became incredible at infiltrating deep into the tumor core. They even produced elevated levels of interferon gamma.
Which is what?
Oh, interferon gamma is a powerful weapon used by T cells to destroy cancer cells.
So knocking out CD44 made them super soldiers, but SPAC-seq is all about the why. What was physically stopping the normal T cells from doing this in the first place?
Right. By analyzing the spatial neighborhoods, the algorithm looked at what the CD44 receptor on the T cell was physically touching in its immediate environment. It identified a corresponding ligand called SPP1.
SPP1.
Crucially, this SPP1 molecule was being pumped out by tumor-associated macrophages. These are a type of immune cell that the tumor actively corrupts to act as bodyguards.
Okay, let me make sure I have this biological dynamic right. The corrupted macrophages are essentially laying down a localized trap in the tissue by secreting this SPP1 molecule. And the CD44 receptor on our T cells is acting like the Velcro that gets them stuck to the trap.
That is exactly what happens. When the T cell's CD44 Velcro binds to the macrophage's SPP1 trap, the T cell gets physically anchored in that specific neighborhood.
And it can't move.
Nope. Furthermore, the spatial data showed this interaction actively exhausts the T cell, completely shutting down its ability to fight.
So it's stuck and exhausted.
Yep. By using CRISPR to knock out CD44, the researchers effectively removed the Velcro from the T cell's boots. Suddenly, it could march right past the suppressive macrophages, breach the tumor core, and do its job.
That is unbelievable. And did this hold up outside of a mouse model? Like, did they test human tissue?
Yes, they did. They validated this exact spatial interaction in human colon cancer data sets using a completely separate technology.
Right.
They saw the exact same architecture in human patients. SPP1 positive macrophages forming a spatial barrier, physically surrounding and suppressing the T cells.
Wow. This raises an important question, though. If the SPP1 CD44 axis is literally trapping T cells in the wrong neighborhood, does this mean that previous clinical trials for immunotherapies might have failed, not because the drug was molecularly weak, but because it just failed spatially?
That is the exact paradigm shift this paper forces us to confront. A T cell might be perfectly armed, but if it gets stuck in traffic miles from the tumor, it is functionally useless.
Which naturally makes me wonder, if spatial mapping is showing us all these hidden traps, does that mean some of our older traditional CRISPR screens, the blender method we talked about, were they just flat out wrong?
Unfortunately, yes. The paper highlights a stunning example of this regarding a transcription factor gene called BUL40.
BUL40?
Yeah. Prior to this study, the scientific community had used traditional bulk CRISPR screens to study this specific gene. And the bulk data confidently stated that knocking out BUL40 was fantastic for anti-tumor immunity.
Because when they ground up the tissue in the blender, the bulk data saw that these knockout T cells were surviving in huge numbers, right?
Right. In a blender, high survival usually equals success. But when SPAC-seq mapped this spatially, it completely flipped the finding on its head. Knocking out BUL40 didn't help the T cells fight the tumor at all.
Wait, really?
Spatially, it caused the T cells to get geographically stranded in the fibrotic stroma.
The fibrotic stroma, that is the dense, restrictive layer of scar tissue that wraps around the outside of a tumor, right?
Exactly. The core of a tumor, the parenchyma, is a highly toxic, highly acidic wasteland. T cells that actually enter the core often die very quickly while fighting the cancer. But these BUL40 knockout T cells never made it inside.
Oh, I see where this is going.
They were stuck on the outer edges, in the safer scar tissue. Because they were avoiding the deadly core, they survived much longer and multiplied.
Oh, wow. So in a bulk DNA blender, their high survival rate looked like a massive victory. But spatially, they were just watching from the sidelines, completely useless against the actual disease.
Precisely. It was a massive false positive. A cell's intrinsic genetic state is inextricably linked to its environmental chemotaxis, which is its ability to physically navigate through complex tissue geometry. Without that spatial map, we misinterpreted a cellular stagnation as a biological win. Spatial context literally changes everything.
That is staggering. Before we move to the big picture, we always want to maintain scientific rigor here. This is clearly a massive leap, but what are the current limitations of the SPAC-seq technology? I mean, it can't be perfect yet.
No, it's not. The authors are very transparent about the current bottlenecks. First, the gene knockout efficiency of the SPAC-seq retrovirus sits at around 75%.
Okay, 75%.
That is solid, but it's slightly lower than some older non-spatial systems that use dual guides to guarantee a cut. SPAC-seq sacrifices a tiny bit of cutting efficiency to ensure that perfect spatial barcode is created.
Got it. What else?
Second, sequencing depth is an ongoing challenge. We are fundamentally limited by how many physical RNA molecules we can capture from a single cell on a glass slide. The algorithmic smoothing in TARDIS helps fill those gaps beautifully, but deeper physical sequencing technology will definitely be needed to push this to its absolute limit.
Okay, let's do a quick final synthesis of the mechanics we've uncovered today before we wrap up. Here is recap number 3. We learned that the SPP1-CD44 interaction acts as a physical, localized trap like Velcro, preventing our immune cells from reaching a tumor.
Yep.
We learned that knocking out Bol-H40 strands T cells in the tumor's outer scar tissue. And finally, we learned that bulk CRISPR screens can be highly misleading without spatial context.
That sums it up perfectly.
Yeah.
If we connect all of this to the bigger picture, what Zhang and his colleagues have achieved is monumental. They have given us a robust mathematical and biological framework to understand functional genomics at a single-cell spatial resolution.
It's a whole new world.
It really is. We are no longer studying what cells do in a vacuum. We are studying tissue ecology. We are seeing the forest, the individual trees, and the root systems dynamically interacting, all at the exact same time.
So what does this all mean? It means the architecture of life is being laid completely bare. And that brings me to a final provocative thought for you, the listener, to mull over as we end this deep dive.
I love these.
Think about this. If we can now map exactly how a single gene edit dictates where a cell lives, who it talks to, and what neighborhoods it actively avoids, could we eventually program our immune cells to act as microscopic architects?
Oh, wow.
Imagine not just sending a drug into the body to fight a disease, but specifically editing T cells so they physically build custom, impenetrable defense structures exactly where we need them, forcing the entire tissue ecology to bend to our will.
Building fortresses inside the tumor. That is incredible.
Right. Think about how seeing the where and the why might fundamentally alter how you understand your own biology. Thank you for joining us on this deep dive into the hidden maps inside us all. Keep wondering and keep exploring.